Hello, hi. My name is Pan Zheng. So I work in OncoImmune. I'm the chief medical officer in OncoImmune. This is a company located in the Rockville, Maryland. I'm also a faculty at the Institute of Human Virology in University of Maryland. So I just want to get started with this title is CD24Fc. This is we are carrying out a phase 3 clinical trial. This is a non-antiviral biological immunomodulator for the COVID-19 therapy. So at the very beginning, not that many people familiar with the CD24 and this Siglet. So this is actually a pathway that the our CEO, Dr. Yang Liu, discovered in 2009, that this is a very important checkpoint in the innate immunity. So this CD24 and the Siglet actually recognize a lot of this danger-associated molecular patterns. When cell death, a lot of the content inside the cell actually release into the outside, and the lymphocyte actually can recognize all these signals, these molecules inside the cells. And these molecules also can have an immune response. So CD24 Siglet is an important, is an innate immunity checkpoint to control the host response to these danger-associated molecular patterns. So this pathway also have many other functions, like a T cell activation, like phagocytosis, and also this leukemia relapse. So today, but I'm going to more focus on this inflammation part. So during this inflammation, so CD24 is a GPI linkered molecule. So it's mostly have a lot of this extracellular domain, and then with the insert into the cell membrane. So the CD24 itself doesn't have the intracellular domain inside the cell. So how do they give the signal? The signal is actually going through this Siglet families. Here's the example is the Siglet G or the Siglet 10. So these molecules have a lot of this intracellular domain. They are actually have this ITIM domain, this inhibitory protein kinase, and they bind to the SHIP1 and give a very negative signal for the NF-kappa B activation. So as everybody know, the NF-kappa B control multiple this cytokine, chemokine, inflammatory factors activation transcriptions. So CD24Fc is a product of the OncoImmune company. So this is a CD24 fusion protein is made by the CD24 extracellular domain fused by the human IgG Fc domain and make it soluble. So you can inject them IV infusion as a drug to human. So there's the mode of the action, the mechanism of action really have twofold. Number one is the CD24 directly bind to a lot of these danger molecules. Then they can reduce the toll-like receptor ligands. So there's a less molecule will bind to the toll-like receptor to activate the immune response. Then CD24 also bind to the Siglet G, Siglet 10 to give a negative signal, directly inhibit the NF-kappa B activation. So we have been doing this laboratory research for quite a long time for Dr. Yang Liu, who already working on this molecule for 30 years. So we have found this is a very important modulator for the inflammation, and then inflammation itself also control the majority of the disease process. So we already have many of these preclinical data showing this in GVHD, in the HIV, metabolic syndrome, autoimmune disease, and this immunotherapy-related adverse event, and the chemotherapy, radiation therapy, protection. So there's a lot of things we are doing. And for the clinical part, we already complete the phase 1 in healthy volunteer, and then 2 phase 2 clinical trial in the GVHD prevention. This is graft versus host disease. When leukemia patient have this, the cure method is really bone marrow transplantation. When they have the transplantation, the graft versus host disease is a major issue and cause a lot of transplantation-related mortality. But today we are trying to focus on the COVID-19. So we think this CD24Fc, especially this innate immunity checkpoint, really target on the very, very key features of the COVID-19 pathogenesis. But this part, including this lung inflammation, so this is pretty much knowing that this causes pneumonia, the major effect is in the lung. But then there's also other things. Then there's a lymphopenia. There's a lot of patients come in, the lymphocyte numbers really, really low. And then there's a cytokine release. The cytokine release is slightly different than the CAR T treatment with the cytokine storm. The number is not that high, but the number is increased, and a lot of cytokine increase. And then the other important feature is coagulation. There's an abnormality on the coagulation. Many young people have sudden death with a stroke, with heart attack. And this is also a very important part of this COVID-19. So we're talking about that one by one. For lung inflammation, why we say the CD24Fc can work. So we have this monkey data. So this is what the non-human primate data. We're actually trying to use this SIV. So very similar to HIV, the SIV also infect the monkey, cause a lot of this AIDS symptoms. So in this experiment, we infect the monkey with this SIV. And after a certain time, they start to show this diarrhea, this kind of lost weight. Then we separate them into 2 groups. One is have a controlled normal saline IV infusion. The other is treated with a CD24Fc with a 3 treatment. So as you see from this, the panel B with all this histology, so on the saline treatment group, the 5 out of 6 monkey really have this very severe lung pathology. They have a very thickened wall and a very thickened fibrosis formation, and it's impossible to have air exchange for oxygen exchange. But then on the right-hand side, there's a CD24, there's 6 monkey over there. 2 got sick, but 4 of them is actually pretty much this normal alveolar walls. So we can reduce this viral pneumonia from 87% to 33%. And when we talk about lymphopenia, lymphopenia is a very important feature associated with COVID-19. And then the other viral disease with that feature is really the HIV, the AIDS virus. So this is the paper, the figure coming from the paper, coming from this Wuhan, when they compare the healthy people with people with COVID, whether they are not in the ICU or in the ICU, the common feature is there's a dramatic decrease in the total T cells, including the CD4 T cells, CD8 T cells. So this lymphopenia is a very, very common feature. We have the experiment in the humanized mice. So all these lymphocytes, these myeloid cells are all coming from human. They put into the immunodeficient mice to make its immune system is human. So this is a humanized mice. Then you can infect the humanized mice with HIV virus directly. So when we look at this panel, especially the C and the D, so this is when we infect the humanized mice, then later on treat with CD24Fc. So this R3A is a virus name, then this have a lymphocyte decrease. But then when you're adding the CD24Fc, the lymphocyte is not dead. They are maintaining the same normal levels. And another important feature is really there's a lot of T cell exhaustion, even though the T cell not dead in the COVID patient, the T cell is exhausted. They have very high PD-1 expression that already shown in several papers. So here in these HIV models, we are showing that with the CD24Fc, we actually can lower the PD-1 markers in both CD4 and CD8 T cells. The CD24Fc can prevent the T cell death and to prevent the T cell exhaustion. Another feature is the cytokine. So this cytokine release syndrome also contributes a lot of this pathogenesis of COVID-19. So this is from two papers showing this disease progression, different date in the COVID, showing the increase of the interleukin-6, interleukin-10, 2, interleukin-4, TNF, and the INF, the interferon gamma. And then the other one is really with the severe case and the moderate case, the severe case showing higher cytokines expression. So this is actually the data coming from the CD25C treated with a healthy volunteer. So people is not sick. So from the blood, you cannot detect any major change because everything is in the normal range. But then we have the patient's blood sample, the peripheral blood cells before the treatment. So day 0 before the treatment, and then the day 3 after the treatment, single dose. Then we do the RNA-seq. From the top left-hand side, is actually see majority of the genes have downregulation in the RNA expression. And many of this significant downregulation is on the cytokine, chemokines. And on this heat map, you're just looking at the shape of this. So from the heat map, the first 4 is from the day 0. And then the other 4 is from the day 3. So the first 4 is really a lot of this red, orange, and yellow. But then when you come to the right part 4, there's a lot of blue. There's a lot of downregulation decrease. So they're including this interleukin-6, a lot of this chemokine CCL, and also the downregulation of the toll-like receptors. And then many of these transcription factors that control the cytokine expression also downregulated. So we actually did the real-time PCR and showing indeed each individually, there's a TNF-alpha, IL-6, the chemokines, they all have significant decrease 3 days after the CD24Fc treatment. Then we go to the last part, the coagulation. So this is the figure showing this deep vein thrombosis in the patients with COVID-19. This is just a hospital admission and at 48 hours. And this a lot of this already have like 79% this have this different degree of the deep vein thrombosis. So this is a very, very important feature. So right now, there seem to be the mortality seem to be decreased in the hospitalized patient. It's really, we recognize this feature, start to give the patient the heparin, the other, this reduce the thrombosis. So this is same data set coming from the RNA-seq, from the healthy volunteer phase 1 data. We can see there's 6 different genes. They are important to regulate coagulation. And in our data set, we show the significant decrease. So CD24Fc also reduce this coagulation-related gene expression. So we think this CD24Fc can really target the most important esogenesis features of this COVID-19. They target on the lung inflammation, they target on the T cell loss and the exhaustion, they target on the cytokine release syndrome, they target on the thrombosis process. So we designed this phase 3 clinical trial. with very simple, just make it a randomized, double-blind, placebo-controlled trial. So this is a phase 3 clinical trial. We plan to have 230 patients, adult patients. They all have a severe or critical COVID-19. And we one-to-one randomize them. Half of them receive CD24Fc, half of them receive the placebo. The treatment is very simple, just on day one, single dose, CD24Fc, 480 mg, and IV infusion for 60 minutes. So study duration, 28 days. The primary endpoint is time to improvement. So it's exactly the same as a remdesivir clinical trial. So these are very busy slides. We include the inclusion criteria, exclusion criteria. So important part is really want to show this NIAID 8-point scale. So 1 is death, 2 is on the intubated, on the invasive mechanical ventilator. And our patient population is this 3 and 4. The patients hospitalized on the high flow oxygen device or the non-invasive ventilations. And all the patients require to supplement oxygen. So they are saturation oxygen level have to be below the 94%. So the primary objective is really just compare this CD24 treatment group with the control group, the placebo group. Which one's doing better? Which one can get patient discharged from the hospital quicker? Which one have the lower mortality? And the primary endpoint is this time to improvement in the clinical status from this scale 3 or 4 to upgrade into the scale 5 or higher. So scale 5 is really, you still, even though you're still hospitalized, then you don't need oxygen support anymore. So the major secondary endpoint is really this overall mortality rate at the day 28, and also on day 28, patients either die or still in the ventilator. So we currently now we already enrolled more than 120 patients, so already halfway through. So this is the very beginning with the first 34 patients. We summarize their demographic and clinical characteristics. So the median age is 50, and there's 70% is from the Hispanic. So a lot of this relative younger patients, they are these essential workers. They still go to work. So these patients have a much, much higher risk will get the COVID-19 disease. But we also have a big group of the patients really on the higher end, age over 65. So the patient also have many other coexisting conditions, diabetes, higher lipid, hypertension. So quite interesting that we really have this cancer patients also quite a lot of this COVID-19. And the median duration of the symptom onset to the first dose of CD24Fc treatment is 10 days. So right now we have 120 subjects consent, 117 randomized, and 113 dosed. So in this study, we have 2 interim analyses. One is the first 70 patients to do the futility analysis. And the second interim will be when we achieve the 146 event, we'll do the second interim analysis. So right now, we just have the first 70 patients reach the 28 days. So we are going to do this interim analysis. There's a lot of this data cleaning to get everything ready for this analysis. But very interesting is that we, from all the patients we have been dosed, we never see any infusion reaction. Remember, this is a protein drug. We never see any infusion reaction. We don't see any drug-related adverse event. The most important thing that we can see immediately is this extremely low mortality rate. So this first 70 patients, we have 0 7-day mortality. We have 3 patients, so this come to the 4%. of the 14-day mortality. And we have 7 patients pass away during this 28 days. So it's a 10%, 28-day mortality. So remember, this is still the blinded data. So only half of the patients using the CD24Fc, half patient is a regular supportive care. So we have to put everything into the content. So we need to know what's the usual, your control group, what's the historical control. So even though this historical is only 6 months, but still this Oxford recovery trial is really have the largest number of a patient that we can be used as our the historical control. So in this trial, we have been lauded dexamethasone is really work. They reduce the mortality. So they have this panel A is all participant, and then panel B is a no oxygen support. So that's not our patient. Then there's oxygen only. And then there's invasive mechanical ventilation. The panel D is also not our patient. So our patient is really this panel C, the oxygen only. So in this case, I blow it up and also overlay our mortality rate figures. So on this little bit red and the gray color, there's a usual care, dexamethasone. When we look at the day 14, so the usual care is about 21%. And with the dexamethasone, they coming down to 18%. But then in this even blinded data, CD24Fc with the placebo blinded data, we have a 4% at day 14 and 10% on day 28. So this is a really dramatic decrease of the mortality rate. Let's compare with remdesivir. That's another successful drug already emergency authorization use in US. So this remdesivir have 2 New England Journal of Medicine papers. One is NIAID remdesivir trial. So this they are using the 14-day mortality. So this placebo 12%, remdesivir 7%. And then there's Gilead, there's own trial, 5-day treatment and a 10-day treatment. They have 14-day mortality. One is 8%, one is 11%. So compare in our group, we have 4%. And in our blinded data set, we have 43% is in this high oxygen flow critical patient. And in remdesivir trial, the 5-day treatment, 10-day treatment, there are 30% critical patients. So our patient have more severe and critical case than the remdesivir trial, but we have a better mortality rate. And when we compare this covalence and plasma data, there's also the dramatic difference. So this is coming from the 20,000 patients using this plasma treatment. And overall, 20,000 patients, they have 8.56% 7-day mortality. And if we just compare the patient without mechanical ventilation, there's 12,000 patients. They have 6% mortality. And in our case, with 70 patients, half of them using CD24Fc, they're 0% at 7 days mortality. So the other treatment is the most lauded, and the US government give $200 million to support either this Sanofi's Cyrolidoma or the Roche tocilizumab clinical trial. They all fail in the phase 3 randomized clinical trial. They don't show the difference. They don't improve the mortality rate. So there's a lot of this retrospective studies. They also have this 13% mortality rate and all different numbers. Then when we compare just blinded data, compare with Cyrolidoma, they compare with this critical case. The patient need a high oxygen support. They have the mortality rate, the high dose Cyrolidoma, 23%. And in our case, our high oxygen group, we have 7% S rate. So at the end, in the summary, we think this CD24Fc really target this very important multiple part of this COVID-19 pathogenesis, including the lung inflammation, including lymphopenia, cytokine release syndrome, and the coagulation abnormality. And we think right now, no drug can address all these 4 challenges at the same time. So we consider this CD24Fc could be the real game changer, can really save life and really make the difference in this COVID-19 pandemic. So I'll end it here. Thank you very much.